It's a pleasure to be here. I've learned a ton from this series in the past, so it's really nice to have an opportunity to contribute to it. So what I'd like to do with this talk is give a sort of broad overview of quantitative trait loci, or QTLs. I've put expression here in parentheses, although most of the talk will be focused on expression QTLs. Many of the concepts are really generally applicable to any kind of molecular QTL. And so in terms of the structure of the talk, I'll start with aspects of data normalization and covariate correction, then get into the details of QTL mapping for cis-QTLs, talk about other QTL types, including transient QTLs, splicing QTLs, conditional independent QTLs, as well as context-dependent QTLs. And as I've mentioned, these concepts are really generally applicable. There's some topics that the talk won't cover just for the sake of time. And one of those aspects, which is really key to these analyses, but basically another talk in itself, is how to perform quality control of the core data types. So both on the genotyping side, as well as the RNA sequencing. It also won't be a sort of step-by-step, really detailed walkthrough of how to do these analyses with code examples, not really more of as a bit of a higher level overview. And it also won't be sort of a discussion of QTL results, although I'll present several examples. These will really be more for illustration purposes. And so before I start, I just want to acknowledge that pretty much everything that I'm going to present is built on work that I did and others did as part of the GTEx consortium, also building on a lot of prior expertise of mapping QTLs in other previous studies. So this is really the result of a large collaboration. And so as most of you are probably familiar with, the large and still growing number of genome-wide association studies conducted over the past decade or so have now revealed over 70,000 associations to common diseases and traits, and a large majority of those are found in non-coding regions of the genome, rendering their interpretation highly challenging. But so the hypothesis is that the mechanism of action of these non-coding variants must at least partially goes through modification of transcription. And so the idea behind quantitative trait loci, and specifically expression quantitative trait loci, is then to identify this relationship between genetic variation and molecular phenotype that's the total level of gene expression or alternative expression of different transcripts of a gene, and I'll get to that later in the talk. And so the general concept is that we want to scan the genome either locally around genes or looking for any variant genome-wide that might affect the expression of a specific gene. And often these effects can be tissue-specific. So what I'm illustrating on this slide is an example where in the lung, for example, we find a strong correlation between genotype and gene expression in the presence of a specific variant, and assuming that there's some cofactor that steers this in the lung, whereas it's absent in the heart where this effect is not observed. And to be able to observe these effects, we need to study large populations. In practice, this means that we generally require at least 70 samples or so to be able to reliably detect these relationships for common genetic variation. And then with the goal is to for each variant look for correlations between genotype and gene expression. And so there's many different types of these relationships we can try to identify. And just to start a little bit with a little bit of terminology. So when we try to identify local genetic regulation of gene expression, we call this cis-EQTLs. And this simply means that we're looking typically in a 1 megabase window on each side of the transcription start side of a gene. We're looking for common genetic variants that affect the expression level of a gene. The other component of this is trans-EQTLs, where we're looking for now distal regulatory variation, and that can be both distal, meaning further away from the gene than that cis window of a megabase, or more robustly when the regulation is coming from a different chromosome, as illustrated in the cartoon here. And in addition to total gene expression level changes, we can make the same observation in terms of splicing. So there could be a genetic variant that switches the expression of a specific isoform of a gene. And this can also be detected in the same cis window as the total expression level, as well as looking for these effects in trans. And so the bulk of this talk is actually really just answering the question, how do we identify which of these relationships are statistically significant, considering that we're doing this for every gene in the expressed genome in the specific tissue or cell context, and genome-wide when we're looking for trans associations. And so I'm just showing two examples that you might be sort of in terms of the visualizations familiar with if you've looked at the QTL data before. Typically for the association of a single variant and phenotype, we visualize these in these types of box or violin plots, where you see in the top example, this sort of very strong relationship between the genotype and the expression level. Also in these locus plots, where we see essentially the several thousand common variants in a genetic locus and their association p-values. We on top of that, sort of illustrated with the LD level between different variants. So variants that are in strong linkage are highlighted in these plots. The second example I'm showing here is much weaker. And so there's a slight trend towards lower expression levels as a function of genotype. And this is also much more ambiguous in this locus visualization. And so the goal of all these analyses is identifying which of these signals are significant. In the top case, the association p-value is so strong that it will survive any sort of genome-wide multiple hypothesis correction, but cases like the signal at the bottom here are much trickier. And so it's really, how do we come up with a robust framework to determine what the significance levels of any of these associations? And so on this slide, I'm just showing sort of a broad map of the workflow. And so starting with the study data consisting of genotypes, RNA-seq data, as well as sample metadata, we essentially, the first couple of steps are to generalize normalized expression matrices, as well as sets of covariates to correct for unwanted variation, and I'll get into this in much more detail. Once we have these inputs, then we can actually perform the various types of QTL mapping, including the discovery of cis-QTLs, trans-QTLs, conditional QTLs, and also context-dependent ones, and I'll get into all of these aspects. And the outputs from that are typically a number of files, one sort of summarizing which genes have significant QTLs, as well as sort of the lead variants for those, and then summary statistics for all associations that have been found. And then using those results, there's many downstream analyses that can be performed, and that's including replication, fine mapping, and so on. And so the focus of the talk again is really sort of on the central part here. There's been there's other very nice resources for some of these downstream analyses, including in this primer series. And so if there's no questions so far, I'll sort of start with the first section, just giving an overview of data normalization and covariate correction aspects. So just very briefly, partly also because we've developed tools for this, there's in terms of the RNA-seq quality control, it's really important to sort of remove obvious outlier samples that might indicate poor quality, or identify sample swaps. And in any sort of large scale study, like this, that this will invariably happen, that some of the samples might get swapped somewhere in the process, and identifying those is critical to avoid just loss of power because of misassigned samples. And so if you're interested in this particular topic, feel free to get in touch with me, and that I can provide more details. The first sort of really critical step, then, once we have the RNA-seq data in hand, and that means essentially just a sort of a quantification of read counts for every gene for every sample. The first critical step is to apply normalization between these samples, because as if you're familiar with RNA-seq data, it's really sort of, it's a relative measurement, and if you conduct the experiment for several samples, there's no normalization between these samples. The only sort of normalization factor is the library size, meaning the total number of read counts that you get for a specific sample, and that just experimentally can be variable. And some of the standard normalizations will just sort of normalize the scale to units called transcripts per million, which is sort of an absolute scale, and ignores potential outlier effects. And so it's important to actually have a normalization that mitigates these. And there's several methods for this. And some of the best performing ones are called TMM or size factors from DSeq. And the idea there is to normalize all the samples that you're using to either a representative sample that represents the median, or construct such a representative sample from the set, and then identifying a scaling factor that rescales the normalizations relative to this. And here, just illustrating why this is important, is the relationship between just raw library size, so the total read coverage, and the scaling factor. And while the correlation is fairly high and the scaling factor is close to 1 for most samples, there are some clear deviations from this. And so making sure that these are well corrected for is important. And in terms of some of these well-performing approaches, there's really relatively little difference between them. So any of those would in practice be reasonable to use. And so in terms of this normalization, this can be applied to samples that are largely similar or have weak perturbation between them. But I don't want to give the impression that this between sample normalization will enable comparing and combining samples from vastly different tissues or experimental contexts. And there's this really nice review from maybe a few years back that really highlights these issues. But the general idea is that once the deviation and the perturbation of the transcriptome becomes too strong, these sort of computational normalization approaches, which rely on having at least a median range of gene expression that's unperturbed between samples, that falls apart, and then the normalization methods also start to fail. And so after normalizing the samples, the next important steps is identifying potential confounding effects. And there are
Oh, sorry to interrupt. There was one question that came up based on your last statement, saying that EQTL is mainly for non-coding SNPs. And does that make EQTL and PQTL complementary to each other in some sense?
That's a great question, and it's important to clarify that. So what one could say one of the main goals is really to facilitate the interpretation of non-coding SNPs, but because identifying a sort of functional mechanism for those is much harder. But there's definitely coding SNPs that can be both EQTLs or PQTLs. And the analysis in itself, in terms of just the mapping, is actually agnostic to the specific characteristics and functional impact of a variant. So we really take all common variants in a cis window around the gene and look for associations with a molecular phenotype. And if we do this with, we can do this with expression, with gene expression, we can do this with protein levels, and some of those QTLs will be the ones that generate that regulate general expression levels will likely be the same, and so the QTLs that modify that are at the same time sort of modify the protein structure or function might have other downstream effects. But in terms of the detecting which are significant associations between variants and the selected phenotype, the general statistical approach is the same and agnostic to the type of variant. So the next step is really trying to make sure that we're not confounded by unwanted technical or sort of population effects. And you might, if you've worked with sort of genome-wide association studies, many of these concerns are exactly the same. In terms of experimental batch effects, there's many studies that have nicely shown under sort of highly controlled experimental protocols how measurements can be sort of robust or vary between different labs. And here's just an example actually showing that when quantifying just exon level gene expression for samples sequenced at many different centers, the sample identity remains relatively robust, but then when looking at transcript level, there's much more variation and also a little bit of a trend towards center-specific effects. And so that's important to keep in mind that there's really experimental variation can add a lot of variance to gene expression, and that needs to be corrected for when we map QTLs. To a similar degree, population structure, we want to make sure we correct for this, not to be confounded by potential effects that are correlated with population structure. Here's one of these examples of technical effects between centers, and one of the stronger ones is just minor variation in GC content that can arise as part of the library preparation protocols in RNA-seq. And the example here is where there's just in terms of looking at the fragment GC content distribution, there's a bit of variation on the upper end of the GC percentage. But then when we look at individual genes, this can actually translate to quite massive differences in coverage. And so there's in this particular example, the coverage on this exon is almost lost in one of the centers. And this means that now in terms of both global expression levels and potentially splicing differences, we might artifactually believe that this represents a change, even though it's entirely driven by a technical artifact. Similarly, when working with degraded samples, such as some of the postmortem samples from GTEx, the RNA can be at least partially degraded. And in extreme cases, this manifests as a strong 3' bias. So looking at the 3' UTR, since it's based on a poly-A selection protocol, we see a strong enrichment towards this end of the transcript. And that too is important to correct for, especially if there's variability of these effects across samples. We want to make sure we normalize this out. And so there's listing these potential effects individually and trying to correct for them explicitly is really challenging. One, because for a large scale study, there can be many of them. And there's absolutely no certainty that we're capturing or aware of all of them. And so there's frameworks to do this automatically in a sense and try to identify latent factors that best capture this unwanted variation. And so there's two more points that are important to make here, and then one I'll get into more in detail, and that is that usually the strongest contributor to expression variance is actually just the cellular composition heterogeneity of different tissue samples in a study like GTEx. But this can also be the case if just in terms of whole blood or PBMCs or the blood-based samples. There's quite a bit of variability between individuals. And so accounting for that and these various technical factors to try to understand what variance components we're identifying with latent factors, having access to really good sample metadata is very important, so that we can actually then sort of disentangle what's captured by latent factors and assign this to cell type specific variation or technical effects like the ones I've shown before. And here's just an example sort of highlighting how strong the cellular heterogeneity differences can be between samples, shown for both heart tissue and colon in the bottom row here. So this is part of an analysis where we actually looked at the enrichment of specific cell types in these tissues. And I'll just use the bottom example here to highlight where some of these differences are coming from. So just in terms of the tissue sampling for these colon samples, there will be samples where almost all of the tissue collected is from the sort of muscular layer of the colon, whereas other samples might be almost purely epithelial. And so the cell type proportions across these can vary drastically.
Francois, there was a question about why you use PEER for adjusting for latent factors rather than SVA.
So so PEER is essentially just is a sort of Bayesian framework for identifying such latent factors. There's many other approaches that can be used, including principal components. There's in the PEER paper and prior work from the same authors, they show that the PEER framework is more efficient in terms of capturing unwanted variation with fewer components, but I've seen several QTL studies that use principal components, and there's probably not a very drastic difference between these approaches in the end. SVA is designed to capture variation that's orthogonal to a variable of interest, and it's more often used in the context of differential expression. And so I'll talk a little bit about sort of context dependent QTLs later, and SVA is certainly a framework that could be used for these types of analyses. But overall, these different approaches try to do the same thing and are largely compatible. And so here, just sort of to belabor this point of in sort of reassigning and in terms of quality control, the variance captured by by any sort of latent factor that we're selecting, whether it's peers or principal components, what we generally see is that the first couple of these factors are really strongly correlated with the cellular composition of tissue, and what I'm showing here is just sort of an enrichment score, a correlation between an estimate of the tissue heterogeneity and PEER factors, and at the same time these effects are only weakly correlated with known technical covariates. In terms of selecting these PEER factors, what is the optimal number of factors or components that we actually want to remove from the expression? Here it gets a little bit trickier, because on one hand, it's again this idea that we're with metadata that we collect and data that we can sort of orthogonal information we can infer from the sample, such as cell type composition, can only inform us to some degree the correlation between these known aspects and latent factors, but that's no guarantee that we're really removing everything that's a potential confounding effect. And so the strategy that we employ typically is actually select the number of factors that will maximize QTL discovery. And here I'm just showing this from an earlier analysis in GTEx, where these plots show just an increasing number of PEER factors and then the number of genes with at least one significant QTL that we identify. And this is also to some degree sample size dependent. This is a little bit heuristic in terms of how we choose these. Generally, we sort of cut off these curves as they start to plateau. One danger of selecting too many peers, and this will be stronger with principal components because it's an orthogonal decomposition of variance and will just increasingly remove signal. But a bigger concern is when we're using these same factors for discovering cis regulatory variation and trans regulatory variation, is that as we increase the number of peers, they might actually start to capture trans effects. And so one important quality control is actually to test for this. And so what we generally do is we just run a sort of genome-wide scan of the PEER factor, of each PEER factor, to try to see if there's any enrichment for specific loci that are associated with this. And here is just an example showing a case where we don't really detect such effects. so that sort of covers the aspects of data normalization and covariate corrections. And so with this, I'll get into sort of more the details of QTL mapping. The model itself is very simple. So it's just a simple linear regression of genotype onto phenotype with a set of covariates that I've discussed. So these peer factors, genotype principal components, and some technical batches that we may want to include, especially if there were sort of technical batches on the genotyping side. So importantly, typically we don't really care about the coefficients of these covariates, so we just residualize the phenotype and genotype relative to these covariates using the sort of orthogonal projection matrix, which should be pre-computed, and this residualization can be done very efficiently. And so in practice, rather than sort of regressing on these sort of box plots that you typically see, the residualized space generally looks like this. Here's an extreme example, again, of a very strong association.
Francois, there was one question about when you add additional peer factors to maximize the number of EQTLs found, how do you differentiate between adding true positives and false positive discoveries?
Yeah, that's a good point. So the advantage of the peer factors is that if you add too many, they tend to be correlated and will, in most cases, add noise and lead to loss of power rather than false positives. At least that's what we've sort of seen in practice. But I think it's an important point. There's no guarantee that this will never be the case. The sort of best sanity check is really looking for trans signals that might get captured. In which case, we're knowing that we're starting to remove real effects. And so then here, I just wanted to give an intuition of going back to this question of how do we identify a significant signal. I wanted to give an intuition of how this is done. So in a specific locus, we can look for the strongest association between any variant and gene expression. But how do we know that this didn't just arise by chance for this particular gene? And so what we do is just apply a permutation strategy where we permute the sample labels and then retest this and ask under this permutation how often do we actually see a strongest association that's at least as strong as the one we saw with the unpermuted labels. And that's sort of illustrated here. So we typically run a few thousand permutations, and then we employ an approximation strategy that sort of extrapolates from this distribution, which can be modeled with the beta distribution to get a more accurate estimate of the empirical p-value, and use these empirical p-values to determine which genes have at least one significant signal. Here's a couple of examples of this. One case where there's a very sort of localized signal here that's strongly significant after permutation. Another case where there's a lot of LD, so there's essentially in almost 2/3 of the regions there's variants that are strongly correlated that tag the same signal, but this also under the permutation is very significant. And here's a much more marginal association where it's on. The nominal significant association p-value is still relatively strong, but it's even visually in the locus less clear. And then with the empirical p-value, we also see that this is not significant by any means.
Francois, there was a question just about how you actually do the permutation and compute the signal after the permutation's been completed.
So the first scan is in the locus. We have a few thousand variants. We compute the correlation between the genotypes for each variant and the phenotype, and then we pick the lead variant. And this relies on an order on a mapping between phenotype and genotype sample labels. And the permutations are just a scrambling of these labels. And then we perform exactly the same calculation. And so in this plot, the orange line indicates the p-value we had for the non-scrambled labels, and each scrambled p-value contributes to generating this gray histogram, so this distribution. And then we extrapolate this to get more accurate. But it's really by the permutation builds this null distribution. And we do this because this varies highly for you. These are all sort of on the same scale. These distributions vary highly for across genes. We really have to do this for every gene individually. Once we've done this, we can apply FDR for false discovery rate across all genes, and we typically you do this using story Q values, but other corrections such as Benjamini-Hochberg would also be appropriate. And as I mentioned, we do this using these beta approximated empirical p-values. The same sort of framework can be used for a replication. So what this means is typically, and this actually also answers one of the earlier questions about how do we know if we're capturing a true signal or if the addition of peer factors adds false positives. One way, which also has some caveats, but is to look for replication in an independent study. So here I'm just showing examples from GTEx where for tissues that had a paired tissue type in the Twins UK study, we wanted to see how the p-values from GTEx replicate in those. And what we're looking here is essentially so assuming that the signals are discovered in GTEx are real, now what we want to see is enrichment of small p-values for all these gene variant pairs. And that's what these histograms are showing. So the histograms are showing these replication p-values, which tend to all be very small. So just visually here, less than the sort of nominal threshold of less than 0.05. But to quantify this, we can use the same strategy behind the Q values, which is sort of to estimate the proportion of true positives. Now linking this back, both sort of in terms of the FDR thresholds and the permutation scheme to nominal p-values. Is it actually, I've shown you examples showing that these empirical p-values can vary drastically between different genes, but in practice, we often get asked, like, how does this relate to actually, if we calculate a nominal p-value threshold based on that, couldn't we just use this as a sort of general cutoff for genes to avoid having to do all these permutations? And the answer to this is sort of shown in this graph, and it's essentially no, because there's quite a bit of variability especially for a small number of sort of outlier genes in this space that have really very different cutoffs corresponding to a set FDR level. And this has a little bit of an association with sample size, which is sort of increasingly here, but only vaguely. Everything I've described so far in terms of the mapping relies on the normalized gene expression space that is described earlier. And while this is great for performing the associations robustly, the effect sizes that come out of this are not necessarily meaningful or interpretable. And so what we actually want to know and quantify is the relative sort of cis-regulatory effect of the alternative allele compared to a reference. And the way we quantify this is as allelic fold change, which is the definition of this is shown here. So it's essentially the log fold change of expression of the alternative allele over the reference. And this means now that to compute this, we actually have to work with sort of the untransformed gene expression data. So just with the sort of the read counts that have been corrected for between sample variation, but without further normalization. And this then means that we are essentially dealing with a model that has now multiplicative noise instead of the additive noise of the simple regression model. And to solve this, we essentially, we can apply iterative approaches, but these are not necessarily tractable for the large number of associations that we want to compute this for. And there's now efficient sort of linear approximations to speed this up. And here's just an example of what we can learn when we have these allelic fold change effect sizes in hand, and one of these, the important takeaways is that as we increase sample size in QTL studies, we tend to discover increasingly small effects. So the discovery of large effects saturates with a few hundred samples, and then as sample size increase, we detect increasingly smaller effects. So just briefly now, after sort of going over the bulk of cis-QTL discoveries, I just want to mention a few points about other types of QTLs. One is splicing QTLs. So instead of just quantifying total read counts for every gene, we can quantify splicing phenotypes. And one really nice approach is described in the paper describing the leafcutter method, which just computes intron excision ratios and groups them as sort of connected components. So groups of introns that share pairs of junctions essentially get clustered together and quantified together. And then these ratios get computed for all of these clusters within a gene. And in terms of actually mapping QTLs for these, it's not fundamentally different from the expression QTLs, but we want to do this as a sort of group phenotype, because to identify whether there's a significant splicing QTL at the gene level, we're now essentially testing each of these clusters for the gene, and since we're always picking the smallest p-value, we want to make sure that in this case, now that we're testing multiple phenotypes for each gene, we're correcting for the selection of a smallest p-value among n phenotypes, also as part of the permutation scheme. And that's really all that's different for the splicing QTL discovery. Without going much into detail here, an important aspect of splicing QTLs is that there's now sort of a more subtle technical effect that can happen during the RNA-seq alignment, which is called allelic mapping bias. So if a variant, if a specific RNA-seq read contains a variant allele, this might actually mean that it aligns to a different place in the genome than if it contained the reference allele. And this is a generally weak bias, but specifically if it affects the splice site, this could actually induce a false positive splicing QTL and needs to be corrected for. So just something to keep in mind if you're interested in splicing QTL analyses.
So there was a question actually about that and the kind of grouping and clustering that you observe with the splicing QTLs. I guess, do you have any more insights into how they cluster or what the biology behind it might be?
So this is not really sort of biological. This is essentially just a computational strategy to robustly identify these associations, to make sure that essentially that we're not, because the main risk when you're testing now multiple of these groupings per gene is that you're just sort of inflating the p-values by selecting the smallest among many. But the biological interpretation here is more complex. So essentially what comes out of this analysis is a SNP that's or a set of variants that are associated with alternative sort of exon or transcript usage within the patterns detected by this method. So it does miss some alternative splicing phenotypes, such as alternative UTRs and poly-A-tails and so on. But within that framework, this will detect changes. But then to identify the specific change, one needs to go back often to the raw data and sort of isoform assembly data to figure out what the switch exactly corresponded to. So just very briefly about trans-eQTLs. So in a way, the detecting trans-eQTLs is simpler than cis, because just performing the association scan, we're now looking for a correlation between a specific phenotype. This can again be a splicing phenotype or just gene expression and all variants across the genome. And so here it's just more about computational efficiency and being able to conduct these scans. But with studies, sample sizes such as GTEx, we're actually still very underpowered to detect these effects. So we only detected a few dozen of these trans E genes in GTEx. And there's alternative strategies where, such as the one employing EQTL gen, because of this more limited power, one can then just, for example, take trait-associated variants and specifically test those for trans effects. The reason I wanted to quickly bring up some details about trans effects is that there's, if you're interested in conducting this type of association, there's an important technical confounder that needs to be corrected for. And I'll break for a question just after this. So there's what can happen is that there's between two genes, there might be a region of homology, and then reads that essentially originate from one gene might erroneously map to another. And in the case of trying to detect trans QTLs, this means that a cis regulatory variant that affects a gene that contains such a region might then actually just sort of artificially drive a trans association, just because the reads from the cis gene map to the trans gene, and that association gets picked up. And here's an example that shows what's in the cartoon. There's a region with low mapability in cis, which has this, you can sort of see the difference in colors in coverage, and then that translates to the trans.
Sorry, thanks, Francois. Everyone's very interested in your approaches. And so the question was just, when looking at these trans EQTLs, what's the relative power you need to detect a trans EQTL versus a cis EQTL in terms of sample size?
I don't have a good answer for this. I actually don't have this plot in my slides. I thought, but sort of, I think sort of the first figure of a typical QTL paper, especially from GTEx, usually shows the correlation between sample size and number of E genes discovered, which sort of shows these growing and now plateauing curves. We didn't observe something like this in trans. So there's a slight indication that this starts to grow for the two tissues highlighted here are thyroid and skeletal muscle, which are some of the tissues with largest sample sizes. But it's difficult from this to extrapolate from this how far we'll really need to go to detect these effects. And there's other confounders that we're just starting to explore. Many of these trans QTLs could actually be driven by cellular composition effects. So this is definitely something that's still being worked on, and hopefully as larger scale studies get done, we'll learn a lot more about these and be able to discover a lot more of these effects. So just being aware of the time, one more type of QTL mapping that I wanted to describe is conditionally independent QTLs. And how to detect sort of allelic heterogeneity, because for many genes, by applying this type of conditional analysis, we're able to detect many different independent signals. Here's just sort of an extreme example of this, where at the top row, I'm showing the bulk association signal, and then the bottom two rows show a conditional analysis. And just as a reminder, that when we talk about conditional analysis in this context, this means that once we find a sort of lead variant associated with the gene, we can then add this to the covariates, essentially sort of regress out the effect of that variant, and in the residuals now ask, is there another signal that remains that also passes our significance threshold? And so here I'm saying this is an extreme example, because there's really these two LD blocks that are sort of perfectly separated. And so if we regress out the signal from the first, then the second signal remains and is a strong in this as a secondary signal here. What's nice in this particular example is that we were actually then able to map this back to effects originating in different cell types. And so on that note, I just wanted to very briefly give a last section on identifying context-dependent QTLs. And this can mean many things. So the focus of what I'll cover will be sort of trying to identify cell type specific effects, but this could be really any sort of context, meaning this could be differences between genders. It could be dynamic QTL, so responding to a specific stimulus. for example, during development and so on. And so one way to identify these effects is using the interaction models. So now complementing the original simple model where we just look for a correlation between genotype and the phenotype, we're now adding an interaction term and this gene by interaction cross term here with again a set of latent factors and covariates. And so in terms of detecting these effects, it means in terms of just sort of an intuition. So you could imagine for samples where this effect is very weak or not present, you'll essentially see no difference in terms of just total coverage of reads in the RNA-seq between three genotype groups illustrated in gray, blue, and red here. But as this effect becomes stronger, for example, if there's a strong enrichment in samples for a specific cell type, these and the coverage here and the QTL effect will become more apparent. We can also visualize these in these types of scatter plots, where as the cell type enrichment grows, we then see a separation between the different genotypes and the QTL effect becomes apparent. And so we've sort of extensively applied this to try to identify cell type specific effects, but the general idea of trying to identify context-dependent QTLs in this manner was already proposed in this earlier paper by Ludefranke's group. And here's an example of the sort of cartoon I showed in the previous slide, where at the bottom here, if for samples where there's a low proportion of keratinocytes, there's a relatively weak difference in coverage across the axons of this gene for the different genotypes, and this effect becomes much more pronounced with samples with high keratinocytes, and we also see this in the scatter plot here. Why is this important, and why do we think this is a really exciting direction to go in? Is that when we're actually starting to incorporate cell type specific effects, and we're able to find a lot more colocalizations with GWAS signals. And so I'm rushing a little bit here towards the end because I just wanted to sort of give a broad overview of the different types of analyses. But what this talk leaves out is sort of a very important aspect of downstream analysis. So once you have your sort of EQTL results in hand, meaning the different E genes and the conditional independent signals, potentially an important next step in many analyses will be to actually fine map these signals and then find colocalizations with results from other studies, including GWASs. And so here's just an example showing this. So typically what we want to do is if we showing the locus for just an example gene here, when try to identify a QTL that underlies the association that we see with the GWAS signal. And what the example here shows that we're not able to identify such an association, such a colocalization with just the bulk QTL signal. But then when we actually apply these sort of context-dependent analyses and try to identify cell type specific effects, in this case, looking for QTLs that depend on the enrichment of neutrophils, we then actually pick up a QTL signal that colocalizes very well with the GWAS. And when we apply this here across all the blood genes in GTEx, but then also across all tissues, we see a very strong gain in terms of the colocalizations we're able to identify with these context-dependent EQTLs. And indicating that an important direction to pursue here, and others have results along these lines as well, of course, will be to go from these beyond sort of just bulk tissue studies and try to increasingly identify cell type specific effects. Very briefly, sort of all I've shown you before that was sort of conceptual. Here's just sort of a shameless self-promoting plug. We've built this software that allows to conduct these associations extremely efficiently by leveraging GPUs. So TensorQTL that builds on previous work, so it essentially extends what FastQTL, a previously widely used mapper, has been doing, but implements a lot of different modes. Essentially, all the types of QTL mappings that I've described are implemented in this software. So if you have questions about this, feel free to reach out. There's, as I've mentioned, I think that the really exciting the direction for the field now is to go after identifying more cell type and context dependent QTLs. So this is more on sort of the experimental design side of QTL studies, but also increase now sample sizes, especially to identify more trans effects. And there's large-scale biobank efforts such as TopMed, which are generating a lot of multi-omic data, which will provide really exciting opportunities to do these analyses. And at the same time, these new data types and larger study sizes will require improved statistical methods to deal with the challenges that will arise from these data types. And I'll just end here on the slide with different resources to follow up on this. So of course, the GTEx portal, which contains all the results from GTEx and has nice visualizations that allow you to explore the QTLs. A really nice resource, that's the eQTL catalog, which aggregates a lot of QTL studies and makes all the summary statistics available. All the pipelines and software that's behind the analyses and methods that I've described in the talk are publicly available at these two links. And if you have any questions, please reach out and get in touch.
Thank you so much, Francois. That was an excellent and tightly packed review of quantitative trait loci across many different situations. Just as one final question before we end, how do you think this applies to kind of the study of rare variants and its contribution to human disease? So
I've actually glossed over this a little bit. So in terms of the genotype QC at the beginning, these types of studies, depending on sample size, are typically restricted to common variants with minor allele frequency of 1% or higher. And if the sample sizes are small, even 5% or higher. In terms of identifying rare variant effects, I think that for GWAS, there's essentially, and I'm much less familiar with those approaches, but there's sort of aggregate tests that to gain power to identify the effect of rare variants, and that's certainly an idea that I think the eQTL field will go into. So as hopefully sample sizes from biobank scale studies in blood will go towards tens of thousands of samples, those are models that we'll be able to start exploring. Including actually just sort of lowering the minor allele threshold to go towards slightly more rare variants in terms of these standard approaches.